我想跟大家带来一个这个 NAD 检测和成像以及它这个药物研发新机会的这样子一个报告啊。整个报告我就计划用英文来讲了，也方便这个在场的海外的这个学者来理解。The talk that I want to give today is about sensing and imaging of aging-related metabolites. So we believe that these are actually new opportunities for drug development. So we are a team of protein engineers. We do chemical biology by training. What we try to do in the lab is that we want to develop tools so that we can sense the metabolites related with disease and aging, etc. So we have all been through this traditional, conventional blood testing that we need to take a tube of 5 milliliters of blood and send it to a larger machine so that we can do all the screenings and tests. And we always had the dream of a lab-on-chip solution so that we can make many of the testing more available, more at the point of care. So in our lab, we have been developing sensor tools so that this can be achieved. And of course, with the evolution of our technologies, we not only can sense metabolites at the point of care, but also sense the metabolites in live cells, especially in some of the subcellular organelles that can lead us to new insights into diseases. So we have been developing our point of care metabolite monitoring. We developed the fingerstick NAD analyzer. We also have the live cell NAD imaging and live cell NMN imaging that we want to share with you today. So we started the project by asking how to sense small metabolites in blood rapidly and easily. The reason why we want to do this is that for some of the diseases, especially metabolite-related diseases, this monitoring can be important. For example, for the management of phenylketonuria. This is a disease with which the babies would have high levels of phenylalanine, and they have to manage this by taking a low-protein diet. And the parents of these babies have to test their phenylalanine levels using dried blood spots, and this usually take a few days for the parents to receive the results. And during this time, the baby or the family has to adapt the diets based on their experience. What we thought is that we can convert phenylalanine into phenylpyruvate and quantitatively produce NADPH. And once we can quantify the NADPH in a convenient and easy way, then we should be able to quantify phenylalanine and many of the other metabolites using a similar principle. So what we did is we developed we performed the protein engineering and come up with these sensors that contains a chemical ligand, a red fluorophore, and on the other side, a luciferase and a receptor. And this receptor and ligand only interacts with the presence of the cofactor, so here NADPH. And without the cofactor, we have an open sensor that emits blue light. And when we have the cofactor, we have a closed sensor that gives red light. So there is energy transfer in between. We performed several rounds of protein engineering. So in the end, we end up with DHFR inserted by luciferase, and that has a very large affinity change controlled by the NADPH. And then we have the sensor response that's very good so that we can have the point of care sensor. So this is how the sensor looked like. And with an increasing amount of NADPH, the sensor shifted from blue to red. That would allow us to quantify the phenylalanine or the NADPH and hence the phenylalanine level in the samples. In the end, what we had was we take 0.5 microliter of blood and add it into a reaction buffer that conveys phenylalanine into phenylpyruvate and quantified NADPH during the process. So this is how we perform compared to in patient samples compared to gold standard methods such as LC-MS and MS-MS. And we had a very good coherence between the two methods. That gives us sufficient confidence to move to this type of point of care devices so that we can monitor concentrations at a larger scale. So in the end, we had developed this type of bioluminescent sensors and machines to replace essentially the LC-MS for many of the testing scenarios. And the next thing we did was to sense, blood concentrations the drug concentrations in the blood, so that we can avoid some of the side effects. And then papers related to NAD and aging came out. We thought, why not develop a sensor for NAD+? After all, NAD+ is heavily related to the aging process, and many of the clinical-based studies have shown how related NAD is with, for example, here, the physical activities, the sport activities of people. And some other clinical data also showed that this has an implication in, for example, muscle-related diseases. And the first version we had was a semi-synthetic sensor for NAD+. It essentially works the same way. We can see the NAD concentration change and how this is related with the blue to red sensor shift. After having this semi-synthetic sensor, we thought we should make the cost down and make this sensor more available to a broader audience. So we developed the genetically encoded biosensor that's essentially greatly reduced the cost of producing such sensor. So now the sensor is fully genetically encoded. It's produced by E. coli. We can produce a very large batch with a relatively small amount of cells. And this sensor, we can actually tune its affinity between nanomolar to even millimolar concentration ranges. The process, the blood testing process looks like exactly like the glucose meter. So we have just take a 5 microliter of blood and insert it into this machine, and the machine can read out the NAD concentrations. Then we did some second phase clinical trial using the NMN compound. The famous NMN compound actually increases the blood NAD concentrations. This is very much expected, nothing surprising. And we can see there's a dose response with the blood NAD concentrations between the blood NAD and the NMN that's already taken. But we have also noticed is that not all of the people actually respond to the NMN supplementation, and that's, I believe, a common issue in the field, in the industry, that some of the people just do not feel much change after NMN supplementation. And we think that this might be due to a natural in-response existing in the people. And using this sensor, we can actually screen out this group of population and then try to understand why. We also surprisingly found that some of the supplement-naive people, they have naturally high NAD concentrations. And we are performing some of the genetic sequencing on this type of people and see why they have such peculiar or surprisingly high NAD concentrations just by natural, a natural way of living. And of course, we noticed that NAD did decline with age, and for us, it's during the menopausal time that men and women demonstrate different behaviors in terms of NAD concentration change. And before menopausal, men usually have higher NAD than the female population. And even though we have been living with NMN for quite some time now, it's not so obvious how stable or how unstable the NAD concentration is in our human body, because we didn't have a very convenient way to monitor this. And using the point-of-care sensor, however, we monitored for more than 100 days to see how NAD can stay stable or unstable during the observation. And our subject 3, she started to take supplementation, just by herself during this monitoring, and we can indeed see that her concentration of NAD increased, so she is likely to be a responder. What's surprising to us is that we also found that FMT, this fecal microbiome transfer, can affect the NAD metabolism. And I guess for the experts in the field, this is not so surprising, but we see that after the FMT's NAD concentration goes up by quite a lot, and this was achieved by a single transplant of the microbiome, and we're now trying to identify which bacteria are actually responsible for this sustained increase. And let's shift a little bit to the field of clinics or to the medicine. So this is a recent paper published by a Spanish group for the rheumatoid arthritis people. And they found that NAD, because of, I guess, chronic inflammations and all that, the patient NAD indeed is lower than the healthy control. And then they grouped these patients into NAD high people and NAD low people, and then subject these people to the same regime of treatment. That's the anti-TNF alpha treatment. That's classical for the rheumatoid arthritis. And then they count for the response rate. They found that most of the responders are actually NAD high, and most of the non-responders are NAD low. So this leads to the speculation that there might be a disease that we try to call hypoNADemia, that indicates a low NAD in the blood, and that has some implications in the treatment of certain disease, such as RA. And one can easily imagine that we might need to measure the NAD concentrations before we start this anti-TNF alpha treatment, so that we can have a likelihood on how the patient could respond. And this may open up some new therapeutic regimes for these diseases. So to summarize this part, we have bioluminescence sensors for point-of-care analysis. We do protein engineering in the labs, and then we have automated machines so that these tests can be done outside of the lab. And we really try to do this decentralized blood testing for aging-related metabolites and also metabolic diseases. And because with that we have the sensor in the labs, now we can do some high-throughput NAD drug discoveries, because we all know that the current solutions for NAD boosting is somewhat limited, for example, due to IP issues. So you can't really establish a very strong IP on molecules such as NMN or NR. So we're actually looking for these new NAD modulators. So because our sensors are fully genetically encoded, we can put this into different compartments of the cell. For example, here mitochondria and the nuclear. And we did some screenings, and not surprisingly, many of the NAD boosters in our compound library are actually PARP inhibitors, because this inhibit PARPs and increases the NAD. But also, one of the hits that we found is alpha-GPC. What it does is that it decreases the mitochondria NAD, but increases the nuclear NAD. So what we believe that thanks to the availability of NMN or NAD precursors, so tuning the NAD absolute concentration is no longer a challenge. So we shifted to finding modulators that can dictate where the NAD goes in the cells, because we believe that their location in subcellular compartments actually control many of the functions. And that's one of the stories that I will show you later. And of course, in the NAD modulation field, you can find specific targets such as CD38 and NAMPT. So using the sensor, we can easily set up screenings for compounds such as NAMPT agonists. And we did find one that coming from this herb, this Chinese traditional medicine herb. So we have 3 agonists that can boost the NAMPT. What this does is that it helps the secretion of extracellular NAMPT, and it helps the metabolism of lipids in I believe this is the liver cells, and it reduces the triglyceride and also the free fatty acid in the cells. Now we're testing this in obese and also NASH mice models to see how effective these compounds could potentially be. And I just mentioned the subcellular NAD dynamics. So some of the papers has already indicated the location of the NAD metabolism is actually important to determine the fate of certain cell differentiation or certain diseases. So like here, evidence showed that sports or lack of sports decreases NAD, But now I believe that for the drug development field, people are shifting more to the subcellular modulation of NAD metabolism. So one of the examples that we saw was, again, with rheumatoid arthritis. We looked at the different RA data sets and found that a specific enzyme, that's QPRT, it's in the de novo pathway of the NAD metabolism here, the QPRT. We found that QPRT decreases with autoimmune disease. So two independent data sets somehow like demonstrates this on the genetic level. This already is downregulated in our cell models. So when we treat the cells with TNF alpha, we can see the QPRT is the only enzyme that declines in the NAD biosynthesis. The other enzymes all went up, but QPRT went down. Also, our metabolomic studies confirmed this. We can see that this quinolytic acid to tryptophan ratio gets up. So here it really indeed indicates a blockade in the QPRT metabolism. So the detoxification of quinolytic acid is deficient in this disease. So then what we did is that we put the sensors into the live cells and into different organelles and to see how QPRT can impact the NAD homeostasis, if you wish, in the cells. Of course, QPRT is in the NAD biosynthesis. You naturally expect that a lack of QPRT would lead to a lack of NAD. And this is indeed the case. We have seen that the cytosolic NAD went down, ER NAD went down, C-Golgi NAD went down, but surprisingly, we found a hyperinflation of NAD in the trans-Golgi networks. And this has, or if you want, a time-dependent response. So if we treat the cells longer with a silenced QPRT plasmid, then the C-Golgi NAD goes down further, but the trans-Golgi NAD goes up. So we believe that this is likely to be true, and we also imaged some of the trans-Golgi markers, and we found that indeed there is a Golgi expansion during the deficiency of QPRT that's induced by inflammation. And we did some gene sequencing and found that NAD protein, like the ADPR process, the ribosylation process is heavily downregulated. So we were thinking, since we're looking at the C-Golgi, the trans-Golgi NAD inflation, we look at the proteins related to PAR-relation process into in the Golgi network. And we found that PARP12 was responsible for this inflation. So by looking at the subcellular dynamics of NAD metabolism, that can actually allow us to identify downstream effectors of the deficient QPRT, that's the PARP12. And having identified this, we try to come up with therapeutic or rescue methods for the QPRT deficiency. We developed this gene therapy that delivers the QPRT gene into the mice model. And we found that the AAV QPRT has a very good effect on protecting some of the bone erosions during the RA. And many of the clinical scores went down, and so we can delay the onset of the disease, and we can indeed show that here the AAV QPRT downregulates many of the inflammatory cytokines. So this sort of hint to a therapeutic regime, and also hint to a potentially new, well, not necessarily new, but exploitable precursors of NAD supplementation. And we have reason to believe that even though there are three pillars of NAD biosynthesis, they may play different roles, or they have different importance in different biological processes. And the last part, I want to briefly mention that the NMN supplementation, even though it has been around for many years, but do we really understand the NMN supplementations? Actually, there are many unanswered questions with it, despite the wide use of this supplement. For example, this is highlighted in this review, that there has always been an ongoing debate on whether NMN can directly enter the cell, or maybe NR is the more preferred precursor. We actually do not really know that. And some of the work by Imai, he identified this potential transporter of NMN, but there has been always some debates on this. So to elucidate this, we developed the NMN sensor that's specific only to the NMN, not NAD, not NADH, but only NMN. So the sensor essentially works like based on a similar principle that's bred, and then we can do imaging of NMN in the cells. We can also test all the different boosters on cellular NMN. What we then did is we tried to dissect the different mechanisms used by cells. Here is the HEC293 cells to incorporate different precursors. So we have NMN, NR, NRH that enters cells supposedly by different transporters. So we can indeed see that with NMN, the cellular NMN goes up with extracellular NMN. And also here the same. So NR and NRH can increase the cellular NMN. There's no doubt on that. And then if we put inhibitors of the NR transporters, so these are MBTI inhibitors. So the NR uptake of the cells gets killed. So basically you don't increase any the cellular NMN anymore. But the NMN is still effective even with the presence of all these inhibitors. And then we also knock down the NRK, which is responsible for converting the NR into NMN in the cells. Same here. So if we knock down the NRK, the supplementation rates decreased, but nonetheless you can still increase the cellular NMN if you put NMN in the cells. But NR completely is ineffective anymore. So this basically shows that NMN can indeed enter the cells via NR-independent pathways. And lastly, we showed that here the NMN equilibrium in the cells can actually be controlled by many of the other pathways. So it's not just supplementation of NMN that modulates the process, but like subcellular compartments such as peroxisomes and mitochondria, they all have very peculiar NAD homeostasis or NMN homeostasis, and all this can be elucidated using the tool of NMN sensor. And now we're looking into, for example, the role of NAD transporters on the mitochondria and also some of the peroxisome enzymes to see how they can contribute to the disease progression or the process of aging. So with this, I would like to close and thank all the funding agencies and our collaborators, and I'll be happy to take questions. Thank you.
非常感谢这个黎阳教授，这个真是这个青年才俊啊，就是其实比我年轻还几年，所以这个非常厉害，做了这么多很优秀的工作。那现在的话，我把这个话筒就是希望交给有兴趣向这个黎阳教授提问的朋友，请举起你的手啊 ，raise your hand please。那里有一位，麻烦那边有一位。
于老师您好，感谢您的精彩报告。然后上次咱也在线上也简单交流过，刚好今天也有机会聆听您的现场分享，非常的精彩。我这边有一个问题，就是关于 NAD 加的这个，刚刚您也介绍了它对于细胞代谢方面的各方面的机制和很好的一些效果。那我们目前呢，其实像最近关于 NAD 加的临床治疗研究也很多，像比如说在上海王丹阳院士他就在东方医院，然后联合搞了用 NAD 加来辅助癌症病人的治疗，发现可以激活体内的 T 细胞，然后效果非常好。我不知道您有没有去尝试做过一部分的剂量方面的考量。那现在呢，包括像 NAD 加的静脉回输也是抗衰老的一个热点话题。那我们对于 NAD 加单次的普通人或者说健康群体或者亚健康群体乃至癌症群体这种不同的群体，它的静脉给药的剂量是有一个多少的一个建议呢？
对，其实通过静脉给药来补充 NAD 或者 NMN， 其实这种研究更多的是在人的身上做了第一例啊。其实，在动物上面， 直接在静脉中回输 NAD 的这种给药方式还不是特别多见。其实我们看到很多是腹腔给药，或者是这个直接灌胃来给药给这个动物。然后至于说人的 NAD 应该维持在什么样子的窗口才更好，其实这个还缺少更多的这个临床数据来支持。但是我相信，其实比如像我们现在这里的数据，其实是可以看到，不好意思，没有放出来啊，就是像500毫克每天的 NMN 和1000毫克每天的 NMN 是可以显著提高 NAD 水平的。但是像这种提高对于他身体机能的恢复到底有怎样的剂量关系，其实现在的数据还不是非常充分。而我们看到的有一些像 NAD 的静脉注射，从理论计算来讲，这个提供的剂量其实并不是很多。但是也许要看具体的它这个临床实验用的 NMN 的剂量到底是多大，然后如果能够把这个血液中的 NAD 水平和它的这个效果有一个更明显的关联，如果这样的数据可以公开的话，是可以很好的帮助整个学术界来定义这样子一个窗口的。
他当时是王院士他们做临床的时候是用的100毫克，然后连续3天挤药，相当于挤了300毫克。然后不过他是针对于肿瘤病人这种特殊群体嘛，所以我就说想看看咱们这边有没有一些宝贵的经验可以分享一些。
确实我们没有直接去研究那个 NAD 静脉给药的剂量，但是我可以想见，就是比如说它100毫克每天的这个剂量，它发挥的功能，我自己有一个猜测啊，就可能 NAD 这个注射的这种方式给药它利用的 NAD 的调控机制，并不一定是它补充的这个效果，而很有可能是它信号传导的这种效果。
非常感谢。
好的，谢谢于教授。
还有其他朋友 any other questions? Okay, so 我先问一个问题吧，就是刚才您提到就是 NAD 加的这补充的一个方法，就是因为 NAD 目前我们认为它是一个保健品，所以的话我们就是吃。但是刚才你还有 Brian 也提到，就是这个 NAD 当然目前补充来说，就是它肯定是在 age 里面是 decline 的，理论上来说把它补回去应该是有好处的。但是这个补的话，就是除了剂量还有时间，有没有考虑过就像 intermittent fasting 这样，因为它这个跟能量是相关的 NAD 加，对吧？如果假设我们像 intermittent fasting 就是间断性的去补充这个 NAD， 会不会跟持续的补充 NAD 会有一些不同的效果？就是我们在动物上或者在临床上有没有考量说可以设计这样的实验去证明这些方法呢？谢谢。
对的，就是我们对于 NAD 的补充，其实 NAD 补充策略有很多种嘛，对不对？就是我们自己的数据看到，其实日常的生活习惯上对 NAD 调控最大的就是运动本身。如果我们可以经常的做运动的话，自己的身体的 NAD 是天然就会升高的。你也可以想象 NAD 的补充可能是一种对于运动的就是比较简单的代替，对吧？就是一个依从性非常高的，我只要吃了2粒胶囊，至少我在 NAD 的水平上就好像我做过运动一样，对吧？然后当然还有很多其他的补充的方法和策略，但我自己感觉啊，就是可能不代表所有的人的观点，我自己的感觉就是 NAD 补充剂的这个分子本身可能就起到了很大的差别。那可能有的人更需要补 NMN， 有的人更需要 NR， 有的人更需要烟酸等等，这个都是很有可能存在的。那就不一定是说哪一个是适用于所有人最好的。
对，那我觉得这个非常好，就是也是跟您在这个发现，就是说这个 MMN 的那个 transporter 或者其他的 transporter， 就是可能不同人他的 transporter 的 porter 的那个 capacity 是不一样，最后导致它就有差别，对吧？
对，而且其实它背后激活的通路还是很不一样的。我想 NMN、NR、烟酸，甚至其他的烟酰胺或者是色氨酸这样子的，它们所激活的下游的通路和产生的这个代谢的指纹的这个影响是很不一样的。
好，我还有看大家还有问题啊？没有问题，我再问一个小的问题，好不好？就是刚才之前应该是今天上午还是我忘了是 Brian 还是谁提到，就是说这个 MMN 不同厂家这个价格是有很大的差别。就从科研角度上来看来，就是我们摒去他们的质量问题，从科研角度上来看来，这个不同价格的 MMN 其实它有没有一些不一样的东西在里面呢？
从原料的价格上来看，其实 NMN 的原料价格都差不多，就是只是它最后做成这个消费品了之后呢，不同的商家有这个 不同价格定位的考量。就像维生素有很便宜的维生素，也有很贵的维生素一样。但是我们觉得能够影响 NMN 效果的。一个是它本身的这个剂型了，对吧？就是说它本身的这个分子是不是实际的 NMN 的分子？另外可能就是它相应的剂型。我觉得剂型和缓释 formulation 就缓释剂型的这种使用，可能也对 NMN 的身体的效果是有影响的。